Good afternoon, everyone. Thank you for joining BioReperia's webinar on extracellular vesicles in cancer research, new opportunities using Zebrafish tumor xenograft models. A warm welcome to all of you. My name is Zahir, and I am a lab engineer at BioReperia, and I have the pleasure of moderating today's session. Now, I would like to introduce our first keynote speaker, Dr. Linda Bojmar, who is an associate professor at Linköping University. Her research focuses on studying the role of extracellular vesicles in cancer that holds potential for identifying new biomarkers and therapeutic targets. Our second keynote speaker is Dr. Lasse Jensen, co-founder, CTO of BioReperia, is also an associate professor at Linköping University with over 18 years of experience in tumor xenograft models. Dr. Jensen is the inventor of BioReperia's platform. He has developed new disease models within the Zebrafish system, which are now integral to our company's work. He also leads our automation program and technical innovations. Without further ado, I will now pass to Dr. Linda Bojmar.
So thank you so much for the kind introduction. Perfect. So as mentioned, my studies are about cancer and what we nowadays call the systemic effects of cancer, where EVs are important players. And hopefully we can, during this session, also highlight a little bit how they can be used together with the Zebrafish model. Just a few seconds on my background, as mentioned. I have a PhD from Linköping University here in Sweden. My main postdoctoral training were performed in the US at Weill Cornell Medicine in the lab of Dr. David Leiden. And this presentation very much relates to him and his research and the research of the lab, as I did not know much about EVs before I came there. So everything I know is from my time in the lab. And now I'm back here in Linköping, running my own lab, and we are developing the concepts that I learned about during my training. So just a few words about the field, and here I call the whole field cancer metastasis, which I also think relates to the Zebrafish and also to upcoming seminars I know, but this could be a little bit similar for EVs also. So both metastasis and also EVs belongs to this systemic concept of cancer, right? And this is nothing new, even though in recent years we have heard more about this. But much of the credit goes back to Stephen Paget in 1889, who had this theory about not only the seed, such as the tumor, but also the soil needs to be fertile. And with this, we mean today the target organs, so distant in the body, like the liver and the lung, for example. And in 2005, Dr. Leiden coined this concept of the pre-metastatic niche, so changes distantly before the actual overt metastasis occurred. And the question was then, how is this mediated? And here we talk about tumor-derived secreted factors, soluble factors, EVs, as we're going to talk about today, but also others. And I'm also going to touch upon the theory about organotropism and how EVs can be related here. So just a little bit about the concept for those of you who are new to the field. So EVs essentially come from all cells in our body. And this comes with certain complexity, and we also, many might have heard the concept of exosomes. I will refer to this, as I write here, as EVs often, or even EVPs. And the reason for this is some work that I listed here. So EVs are double lipid membrane contained particles that carry cargo, that is protein, nucleic acids, DNA, RNA, lipids, and so forth. So they are like a messenger from the original cell. But however, they are also generated through biogenesis due to several mechanisms in the cell. And this has confused a little bit the nomenclature. Now we often talk about these characteristic EVs as smaller particles within the range of 100 nanometers. If we talk about something below 200 nanometers, we can refer to them as small extracellular vesicles. When it gets larger, we get into microvesicles, apoptotic bodies that might be a little bit different. They are coming from dying cells, for example, whereas EVs are secreted due living mechanisms from the cells. They were originally considered to be dust, platelet dust, as they were discovered, or waste from the cells, waste for the cells to get rid of things early on, 60 years from now. But nowadays we know that they also have active processes, and they are messaging the signaling between cells in the body. And the complexity is also, as I said, that there are many subgroups of these EVs, and that also relates to their size. It relates to what they're carrying, that can be measured by their density. And there are different methods also to isolate these EVs, which is also, it's a new field as such, and it's still developing. So these are also some of the things that one need to consider when we think about EVs. And depending on the question, if it's a more of a mechanistic question where we need to know the cell of origin, even the compartment of the cell, then it might be very important to be very particular with specifically which subset of EVs. If it's a biomarker in a patient, maybe it's not so important exactly where it was coming from or what part from the cell it originates from, as long as it's a good biomarker. So we'll carry that with us as we move forward, and we can take questions on it as well. We did some work during the pandemic to try to summarize a little bit the knowledge we had in the lab, and this relates to the proteome that is carried by the EVs. So we took a lot of samples from humans and from mouse models, and we looked at the proteome by mass spectrometry. And we also looked at some of these compartments where we can find EVs. So naturally, systemically in the body, as I mentioned, so in plasma, in other bodily fluids, they are secreted from cells. So we used cell lines, but also from tissues, which again gives a little bit more complexity because we have more cells there. We have potential also contamination from death and so on. But we're trying to find common denominators, but also differences between tumor types, between normal and cancer, and so forth. And this was published in 2020. So I won't go into any details. I will just say that the message from this was that just by using the EVs in plasma from patients, we could distinguish profiles that separate different cancer types, so to say. So an indication that this could actually be used as a liquid biomarker, as we call it. Mm-hmm. But more work is needed, of course, to confirm this, and this relates also to the work that we're doing today. But a little bit more about the functionality of the EVs then. What we also learned was by looking at these proteomic profiles, there are many molecules that they carry that can interact with the immune system. And this has been summarized in later years by some of my colleagues in the lab, and it's work in progress, but I will tell you a little bit about where we are right now. The pre-metastatic niche, as I mentioned in the beginning, we think is determined by this EV signaling at distant sites, and a little bit the different steps that could potentially be affected by EVs. One thing that I think relates also very much to the Zebrafish model is the vasculature that we know is important when it comes to spread of the primary tumor. So early work when the EV field was still in its infancy was performed by Hector Peinado in the lab. And after David discovered the pre-metastatic niche, as I mentioned, in 2005, they started thinking about what could mediate that. And then at the same time, the EV field developed, so to say, as being active cargo instead of just this waste. So putting that together here is a figure from the early paper showing that EVs, but not just the conditioned media from melanoma tumor cells here, induce vascular leakiness or endothelial cell permeability. So dextran is leaking in the tissues. And this was very early on that supporting this theory of EVs mediating systemic effects related to the primary tumor. And also just another piece from this work showing that indeed inflammation here marked by some of the characteristic genes in the premetastatic niche also induced by tumor growth could indeed be induced just by educating with EVs, so injecting EVs in the animals. And here is some work that was performed when I joined the lab. So I had a little small part of it, but Ayuko Shino, who's now back in Japan running her lab, she showed indeed that you could find these EVs marked in red here in the pre-metastatic niche here in the lung of animals. And she also showed this concept of organotropism. So depending on exactly here, the protein integrin package in the EVs and on the surface of the EVs, and then also subsequently the tumor cells could be directed to different organs in the animals. And this is also illustrated here, was later followed up by another postdoc in the lab, Gonzalo Rodriguez, who showed specifically for brain, where also this blood-brain barrier, specifically the endothelial gaps, could be of interest here, were shown by endothelial cell uptake and the invasion of cells into the brain. So my specific interest is in pancreatic cancer, and also here there was some early work in the lab connecting EVs to the pre-metastatic niche. But my question with a little bit of a translational clinical background was if this also was something happening in humans. And from the initial EV studies, we also wanted to look at the soil. So just illustrated here as the liver, which is the main organ for pancreatic cancer metastasis, and where we from the animal studies knew that the EVs had an effect. We wanted to know if this also happened in humans and if it had a role in metastasis. And this was actually just published in Nature Medicine, so I won't take you through all the data, but basically, again, we saw that in the liver, indeed, there was an inflammatory process going on in the cancer patients compared to controls here. And we could also see that if we labeled for some of the specific immune cells. And I can take questions also, but we know that the liver is important for metabolism, of course, and now we also know that the EVs has a role in regulating this metabolism in the liver, and I will get back to that a little bit as well. To summarize the human studies, we put together the different factors in these pre-metastatic niches, and we were able to create a prediction model for metastatic outcomes. So that's how you actually can use this knowledge of systemic effects of cancer. So processes mediated by EVs. And this is another example also from our lab where this concept of EVs changing distant organs was taken into consideration here by Gang Wang, who was a postdoc in the lab. He showed that EVs indeed regulate directly the liver, and here in terms of the lipid content in the liver, which not only creates this pre-metastatic milieu, but rather here affect the drug metabolizing capacity of the liver, so affects directly some of the cytochromes, for example. And this, I think, also could be very important in terms of drug discovery. So just to end up with, EVs has a lot of potential, I think, when it comes to cancer biology, cancer metastasis, but also in terms of drug development. So we can think about targeting EVs directly. We could go towards the biogenesis from the beginning, the release from the tumor cells. The EVs could be blocked, needs to be designed a little bit specifically towards their cargo. So the protein integrins are good candidates here, but also later on at the side of the uptake in endothelial cells, for example, that lay the line effects, the vascular leakiness and so forth, but also the downstream effects, for example, in this pre-metastatic niche. So I think there's a lot of potential in the sequence of events. And yeah, just ending with a thank you to my team who has been supporting before I think I give the word to Lasse to say a few words.
Yes. Thank you very much. Fascinating data, Linda. Really amazing studies. I will follow up on this with a bit of insights into how the Zebrafish as a model system can be used also for studying EV aspects of cancer biology. So first, a general slide on the Zebrafish tumor model. So what the model consists of is that you implant tumor cells under the skin in zebrafish larvae that are 2 days old, and then you follow them for 3 days to see how the tumor grows and metastasizes. The tumor can be either a simple tumor consisting only of tumor cells, for example, grown as a cell line in the lab, or it can be a complex mix of cells present in tumor material, for example, a patient biopsy that is either enriched for certain cells, if you isolate, for example, lymphocytes and tumor cells and only look at those compartments, or if you just take whatever is there in the same concentrations as they're there in the biopsy. 3 days later, you can then see how the tumor has changed, and this can, for example, be seen as a change in size. Here at the bottom right, you can see how a control tumor usually solidifies and spreads locally, but doesn't grow much. This is mainly because of the short assay period. You don't see a large expansion of the tumor during only these 3 days, but you can still see that the tumor regress if treated effectively with a drug that kill the tumor cells. So then the tumor cells disappear here over the 3-day period. Also, you can see how the tumors metastasize. This is mainly because of the very rapid development of the zebrafish larvae at these early developmental stages, meaning that the blood vessels are expanding during this period of time naturally, and therefore also growing very quickly into a tumor that's implanted. And these blood vessels in and around the primary tumor can then be exploited by the tumor cells to disseminate even as quickly as within 3 days. When it comes to EV research in zebrafish, there is a number of organs relevant for what you mentioned, and also what others have investigated related to EV biology available in the 5-day zebrafish larvae. For example, the zebrafish have at this point developed a quite mature cardiovascular system with a beating heart and blood vessels that are creating a blood-brain barrier in the brain, for example. They're creating fenestrations in the muscle tissue, also a hematopoietic vascular niche here in the caudal region of the tail. So a lot of these specifics related to the vasculature in adult animals are actually already laid down at 5 days and can be studied in these early larvae. Also, other organs such as the liver, the pancreas, the kidney, intestine. I will show what they look like for real soon, not just in an image like this, but they can be identified and followed during these stages of development. The zebrafish larvae is transparent, so you can actually take video recordings of how the EVs are flowing around and being taken up by cells. I will show some examples of this. This is from a paper that I will go into more detail on later by the Goetz group, where they show various ways of EVs moving in and out of the vasculature, either through interendothelial gaps, through transcytosis, or other mechanisms, and also within the tissue, how they're moving in the extracellular matrix and how they're being taken up mainly by macrophages, and this can be patrolling macrophages that are mainly luminal, or it can be tissue-resident macrophages. But the method is similar as shown by this group, the Goetz group. So our data related to trafficking of particles, at BioReperia we have not studied EVs yet, or not trafficking of EVs, but we've studied other particles that you mentioned. And here's an example of 3 different particles that we've studied for a customer. And you can see how at 6 and 24 hours, these 3 different particles exhibited different trafficking profiles, and they accumulated in different organs. Particle 1 here was rather broadly present in both the GI tract, the kidney, the liver, and the heart at 6 hours after injection IV, but at 24 hours, all of the particles were only found in the hematopoietic site. So they had hematopoietic tropism, these particles. These other particles here, they were, on the other hand, broadly found after 24 hours, both in the gills, the heart, the GI tract, and the liver. And the third particle instead had GI tract tropism after 24 hours. So this is, of course, something that could perhaps also be interesting from an EV point of view, to see where do they go? Do they have tropism to a certain organ? And then, of course, understand those mechanisms. So going back to the studies published in 2019 by Goetz Group, this is Hayen et al. They took videos of the labeled EVs that they injected IV in the fish to see how they behave in the vasculature and in the tissue of zebrafish. And they could identify, for example, a mechanism of cellular tissue uptake related to rolling. So they could show that the EVs are docking on the endothelium and then rolling along the endothelium here. You can see that you can take many images per second, basically, because it's a video, and then the speed of rolling and other dynamic details of EV transcytosis in this case, but it could also be other things. I will show you that to the left here, how they're taken up by cells instead. They, however, saw also that at some points the EVs are commonly arresting at the luminal side of the endothelium, and here they're often actually taken up by patrolling macrophages that are also sticking to this area. That can be seen here at the end. You have green macrophages that are taking up these red EVs, and they're often at the luminal side of the vasculature. You can also see that the EVs can go into the endothelial cells themselves, and in this way be moved across the endothelium through transcytosis. But one interesting finding in this paper was that neutrophils do not seem to take up any EVs, at least not in the zebrafish here. They further looked into the mechanisms of uptake, and they found several different mechanisms. For example, you can see EVs that get stuck to filopodia and then via the filopodia move into the soma of the cell and get taken up at the soma. This is a process that they refer to as filopodia surfing, that they characterize through these videos. They can also be taken up either in endosomes or into the cytoplasm itself, and that will then facilitate the different effects that the EVs can have inside the cell. And this is something that goes quite quickly, on average 50 seconds. And of course, the filopodia surfing is a much faster process, as you can imagine from the word surfing, that the EVs are then taking up within a few seconds. So that's, I think, one of the main benefits with the Zebrafish system, that you can really get highly dynamic data that is difficult in other in vivo models, where it's often snapshots at a certain time point. It limits a little bit the analysis you can do compared to the dynamics here. This group was also interested in the role of EVs facilitating tumor metastasis, and they developed a zebrafish melanoma model. So these zebrafish melanoma cells were labeled in red and injected into the fish, and then they injected 2 days later either EVs or beads that were just as an inert control, basically. And they could show that when you injected the EVs from the same tumor cells, they had a pro-tumorigenic effect, both in terms of growth. So the tumor cells that had then experimentally metastasized to the tail region here, they were injected IV, so it's experimental metastasis model, and they were much larger compared to if you injected the beads. And you could also see at day 10, so a bit later, if you wait another 3 days, how they are further growing and disseminating, invading into the tissue here, but not to the same extent without the EVs. So this is perhaps not exactly the seed and soil hypothesis, because the EVs were injected. Oh, they were actually injected early. Sorry, I messed this up. So the EVs were injected first, and then the tumor cells a couple of days later. So this are related to this preparing the soil hypothesis here. But at BioReperia, we have also started looking at EVs and their effect on metastasis. Remember this image that I showed earlier? So we are commonly looking at the growth in this main metastatic region, same as what you saw in the previous slide here in the hematopoietic caudal plexus here. One project we have looked at is EVs generated from tumor cells, osteosarcoma cells, stimulated with different types of loading. So osteosarcoma cells are bone tumors. They are experiencing more or less loading depending on the activity of the individual. And we see that osteosarcoma cells that get very high loading, they release a type of EVs that also mediate metastasis to a much higher extent. Compared to if the loading regime is low or not existing. So the EVs also in our system, so that's a more experimental metastasis system where we actually create a primary tumor and let that metastasize. But also in that system, it seems that EVs, at least under certain conditions, in this case when the tumor cells have experienced high loading, can increase the metastasis to this hematopoietic plexus. We've also tested larger vesicles, if you will, or cell encapsulation techniques. So this is in collaboration with the UK group, Ape Pandit and Vaishali Shurk, who are interested in natural killer cell membranes and putting drug carriers into these membranes. And they have seen that if you only use these gelatin drug carriers alone without the membrane, then they cannot find the tumor cells. But if you put these drug-loaded gelatin spheres into a natural killer cell membrane, then the receptors on the natural killer cells will recognize the tumor cells and get accumulated in the tumor, and in this way target the tumor and release the payload only in the tumor system. So also for that type of studies and the benefit of using EVs as drug carriers, Zebrafish systems, due to their optical transparency and dynamic visualization, can give a different type of data faster and with very high spatial resolution here. So with this, I hope that all of you who came to listen have learned a little bit related to EVs in cancer and how the Zebrafish can help that type of research. Perhaps you want to finish up, Zahir?
Thank you very much. Thank you really very much for a very nice presentation, Dr. Linda Bojmar and Dr. Lasse Jensen. I have got a lot of information from this presentation, and I hope that everybody else have get a lot of information too. Well, we don't have time to answer a lot of questions, but we can take some of the questions. Are there specific characteristics or contents that distinguish cancerous EVs? Can we differentiate between normal EVs? Can we use that as a biomarker maybe? So
yeah, no, definitely, and that's kind of one of the key questions, right? So what we showed a little bit in our proteomic data in 2020 was that definitely there's a different profile, and as I showed, it even separates different cancer types. So yes, definitely related to the cargo, also to the amount. Often, if you look at the cancer cell, it produces much more of these EVs as compared to a normal cell. So just related to the amount of EVs, but definitely also their cargo. And that goes for not only the proteome, but also the RNAs carried and the DNA contents and so forth.
Thanks very much.
Definitely.
Thank you. Another question that we can take: can extracellular vesicles suppress immune response or promote immune evasion by tumors? If, for example, that can, I believe, what does the question is asking if we can stimulate the immune response or suppress this immune response.
Yeah.
With
so
the EVs.
yeah, definitely. So that's one of the lines, is we now know that the EVs interact a lot with the immune system, as you showed, via uptake by macrophages, but also downstream regulators and cells, right? So definitely, and that goes in both ways. I mean, the immune system is a balance, as we know, between the pro and anti-tumorigenic immune systems, for example. So both by the endogenous EVs, they're players in both, I think, but also by engineering, as we mentioned, the EVs, we can also play with these systems and create ways of making more anti-tumor effects, so to say. It goes a little bit towards vaccination even, right? So yeah, definitely there's a lot to explore there as well, I think, when you start manipulating a little bit like you did with the NK cells, I think.
Well, another question to you, Lasse, is what advancement have been made in using zebrafish models to study extracellular vesicles in cancer research?
Yes. So some of the advances was mentioned in the talk here. It relates to labeling of the extracellular vesicles and their imaging inside the fish. It relates to understanding how they're interacting, both with tumor cells and immune cells, endothelial cells, and other cells. There has also been other groups, I didn't show the data, that have shown similar results as I showed with the particles, that certain EVs from certain organs have tropism to other organs. So there's been a lot of data generated with endogenous EVs produced by healthy cells in different zebrafish tissues, what they contain and where they are going, to try and understand the endocrine function of endogenous EVs. And there is a lot of interest these days on the role of EVs in cancer. You saw a few examples of that, but it's really an active area of research now. So I will expect that in the next few years we will see many more exciting findings related to how zebrafish tumor models have been used to better understand the role of EVs in cancer biology.
Sure, of course, yes. Can we take another, probably last question, because we are running out of time? Could EVs be used as a part of immune therapies for cancer treatment? We have talked about this question already, like if you can suppress or stimulate the immune response. So
Yeah. No, but I also think it relates a little bit to the engineering capacity, right? So there's both ways to endogenously engineer the EVs through the cellular pathways, but also exogenous labeling, right? A little bit like you did with the carriers. So I think this makes them versatile, and when we, as we mentioned, when we get towards cancer vaccine or these things, the EVs, the benefit with EVs compared to nanoparticles is that they are natural, right? So one of the problems potentially with nanoparticles is that they induce immune responses that we don't really want to. So I think again it's this balance of inducing a specific response, but not get these unwanted side effects, so to say, by just injecting foreign agents in the body. So yeah, I definitely think there's potential, but there's no more work to be done to exactly pinpoint the different pathways.
Well, thank you very much. I think we already are short of time. Well, if your questions were not answered during the session, or if something comes to mind later, please don't hesitate to reach out to us. We are more than happy to arrange a meeting to discuss your specific projects or any queries that you might have. I would like to thank you, our speakers, once again, Dr. Linda Bojmar and Dr. Lasse Jensen, and our audience who are listening to us today. I wish you all a pleasant evening. Goodbye for now.
Bye. Thank you.
Thank you.